Moving on from bacteria to look briefly at archaea. Again, these are similar to bacteria we've already talked about. They're prokaryotic, so their body sizes and shapes look the same, but they do have profound differences. So dramatically different chemical composition, so the molecules that make up their bodies are very different, and their DNA sequences are also extremely different. And when we look at microorganisms in the environment, it's archaea that tend to live in the most extreme environments. So incredibly high or low temperatures under huge amounts of pressure, so very deep in the ocean, in the deep ice cores, things like that. They can survive in extreme ranges of pH, so acidic and basic solutions, very high salt concentrations. They can live in all of those. In fact, some types of archaea can even live in humans, and they're not pathogenic, so they don't cause disease. They just live in your intestinal tract and will break down or eat some of the tough fibers that our digestive system can't break down. As kind of a byproduct, they give off a methane-based gas and form intestinal gas, which is what causes you to pass gas. It's the bacteria, or sorry, the archaea inside your digestive tract digesting fiber and creating gas, which has to escape and smells awful because it comes from these archaea bacteria. They're also useful in biotech applications. So Thermus equitus is a type of bacteria that we use for doing PCR, polymerase chain reaction, or copying DNA, for lots of biotech applications, from researching new medications all the way through studying forensic cases and getting information for convictions. They also, because of their extreme metabolism, can digest a wide range of things. For example, we use a type of archaea that can digest oil to clean up oil spills. So you spread it over the top of the oil and it digests it down. It doesn't get rid of it completely, but it takes this thick, kind of crude, sticky black oil and breaks it down to something that's much easier to wash away and clean. So there's a wide variety of environments these archaea can live in. Moving on, so we've covered bacteria, we've looked at archaea, we want to look at protists next. And again, these are the first eukaryotic type of cells. So they have a nucleus, and that formed from basically folds in the plasma membrane breaking off inside to surround the DNA. And they also have things like mitochondria that came from symbiotic relationships with other cells. And we've discussed kind of those two concepts in previous chapters. But protists were the first to develop those. And protists are a very interesting and very diverse group. They're eukaryotic, but that's about all they have in common. So the definition of a protist is it's eukaryotic, but it's not an animal, a plant, or a fungus. And we'll look in the next couple of chapters of what an animal, plant, and fungus is. However, these protists cannot be classified into any of those categories because they're missing certain structures or body parts. However, they're eukaryotic. So this is kind of a catch-all for things that we don't know where to classify them. There are animal-like protists that can even be predators. They swim around, they hunt for prey. So they're very animal-like, but they're not technically an animal. They're missing some of those characteristics. There are some that are very plant-like or fungus-like. So plant-like protists, kelp. If anybody's ever seen a kelp forest, I mean, they're huge and they look like these giant plants. However, they're missing some of the body structures that we classify as being plants, and so technically they're not a plant. So we throw them into the protist category. We also have fungus-like protists, such as slime molds, that look like a fungus, but again, they don't have the same body structures as a fungus. So this is one of my favorite definitions, and I oftentimes put this on exams. I might ask, well, what do you need to classify an organism as a protist? And you would say, well, it's eukaryotic, but not an animal, plant, or fungus. So again, this is one of my favorite test questions. Protists can be human parasites. A couple of those, Plasmodium can cause malaria. So malaria is transmitted by mosquitoes, but that protease gets into your body and it infects blood cells and causes them to not function properly anymore. And with malaria, it causes lots of severe side effects. So extreme cramping, it's very painful, kind of high fevers, so you almost hallucinate because of those high fevers. So malaria is a problem in lots of areas with mosquitoes. The Camper's parasite, you can get this easily in contaminated water. Very common among people who are in the outdoors and drink water. This causes nausea, diarrhea, and vomiting, so it's a very uncomfortable parasite to have. Amoebic dysentery is caused by an amoeba, and this is lethal. It causes thousands and thousands of deaths every year. It was also a protease that caused the Irish potato famine, where millions of humans starved to death because their potatoes were eaten in the field by this new invading protease. Viruses, I'll just talk about briefly because we're going to talk about these more in lab. So viruses, again, are non-living parasites. So they don't have a metabolism, they can't reproduce on their own, so technically they're non-living. If you take my genetics class, we'll go a little bit more into viruses and their classification and some of the arguments for classifying them as a living organism and against. So again, they're in that gray area between life and non-life. The only thing a virus is composed of is a caspid, and that's the protein coat, and either DNA or RNA, so some type of genetic material. So a basic virus would just be protein forming a coat and genetic material. That's all you need to be a virus. Some viruses have additional structures. This one pictured here has a phospholipid bilayer and some proteins, and now that bilayer it picks up from its host when it's leaving the host. And the basic infection cycle of a virus, it gets into a host cell by crossing the membrane. It ejects its genetic information, which then takes over the host cell, either viral DNA, or if it's RNA, they copy it back into DNA, takes over the host cell, manufactures more viral parts, and assembles them, kind of like at a car factory, some assembly line. It puts them together and then ships them out of the cell again. So that's the infection cycle of viruses. Some viruses that everybody should be familiar with is the flu virus. And if you've ever had the flu, you know it can be quite uncomfortable. So you're very sick, you can get muscle cramps, all of that. But do you get your flu shot every year? If you answered no to that, you really need to reconsider that and go and get your flu shot every single year. Get them for anybody who's eligible, kids, grandparents. The flu shot is very important because the flu is the single deadliest thing known to humans. It has killed more people than all wars combined. Flu is the deadliest thing that a human could encounter. Even though most people can recover from it, it depends on your particular flu strain. The chances of encountering flu and not surviving are fairly good. So one example of that, there was a global flu that broke out in 1918 through 1919, and it's estimated that 20 to 50, and I've seen estimates much higher than that, 20 to 50 million people at least died in an 18-month period. So out of every 50 people that were infected with this strain of influenza, out of every 50 people, one of them died. And that occurred over an 18-month time period. And this flu didn't behave quite like flus normally do. Normally it's children and the elderly who are susceptible to flus and get sick. However, people from the ages of 18 to 30, they're fairly robust, they have healthy immune systems, they're strong, and they don't get affected by it. However, this flu, the 1918 to 1919 flu, preferentially killed that healthy age group. So from about 18 to 30, those are the people who are dying from this flu. It was the young and healthy people, not the children or the elderly. To put that in perspective, HIV, 40 million have been infected, and only 3 million globally die annually. In 18 months, this flu killed 20 to 50 million people. More people died of that flu in one single year than the 4 years of the bubonic plague that took out a third of Europe in the 1300s. So incredibly, incredibly dangerous. If you want to read a scary book on this, read The Great Influenza. It goes through kind of the story of this particular pandemic. And it also has information on, I guess if you get a newer copy of the book, on H1N1. And H1N1, when it first emerged, was very frightening because this influenza back here of 1918 and 19 was also an H1N1 strain. The difference between the two, first of all, we had vaccines available, so we could vaccinate populations ahead of the flu so it had trouble spreading, so it didn't spread as rapidly as it did in the past, and that kind of contained it a little bit. Also, because so many people had been exposed to and died from this flu in 1918 and 19, and it recurred, after that as well. So many people had died from it that the survivors were the ones passing on the genes for being survivors and having offspring. So all of us are the offspring of the people who survived that flu. So we have some underlying genetic resistance to it as well. So those two factors helped prevent H1N1 from looking just like the 1918 flu did. And there's a video in Blackboard to take a look at a little bit more about this flu. Historically, there's a flu pandemic about every 50 years. So there have been smaller outbreaks in 47 and 76. You've probably all heard of the 2001-3 SARS virus. Some more recently emerged ones, because they're constantly coming out. We're always finding new viruses, is the H3N2 variant. The V stands for variant. And this was first reported in humans in July of 2011, so just a couple years ago. And this virus had been found in pigs in the US in 2010. However, because viruses can swap DNA with other viruses, so if you have two different viruses infecting the same host, they can exchange bits and pieces of DNA and then get a new mix. So basically, this H3N2 virus mixed in one host with the H1N1 virus, and a gene called M, the matrix gene, was exchanged, allowing this H3N2 to infect humans. And cases of this have been found in across the U.S., but including Iowa. So this is one reason I heard on the news recently the state fair is coming up. They recommend that young children and elderly or anybody with a compromised immune system not visit the pig displays, just because they could be carrying this variant of H3N2 and they could become sick with it. You can't tell if a pig has the flu virus, but you could still pick it up if you're in contact with those pigs. So that's been on the news even this year, warning people that when you go to the fair, don't take children or elderly or immunocompromised people into the pig areas. Also, some others, one that we first reported in 2012, this is the Middle East Respiratory Syndrome coronavirus, and this one is scary. If you look at where it's spread to, since it was discovered in the Middle East, there have been cases in France, Italy, Jordan, pretty much all of Europe and the Middle East, and the death rate, so number of cases total has been 77 confirmed cases, number of deaths has been 42. So that's slightly more than 50% of people infected with this virus have died. So this has a very high death rate. 1 in 2 die from this. If you remember when we looked at our 1918 flu, that was 1 in 50. So this is much deadlier than that. Although, we're aware of it and we're monitoring its spread and trying to prevent it from spreading and working on ways to develop vaccines against it. Currently, there aren't any vaccines or treatments for this. H7N9 is another one that emerged in China, first reported in this year, 2013, and its death rate, well, 1 in 5 die from that. So not quite as high as our cornea virus, but certainly higher than what we saw in the previous flu epidemic. So these are all new viruses that could potentially, with just some slight genetic shuffling on the virus's part, become lethal to a majority of the world's population. And again, there's another little video. This is a link to a news story on this H7N9 virus. Other viruses, so oral herpes, or commonly called cold sores, affect 3/4 of all adults in the U.S. And again, this is just a herpes virus, a slightly different strain than what you would find, say, in genital herpes. However, it can be transmitted between the oral area and the genitalia. So genital herpes is also very common and infects about 1 in 6. So out of every 6 people in the U.S., one of those is statistically going to have herpes between the ages of 14 and 49. So incredibly, incredibly common. A couple of other things about viruses. There are 2 types. DNA viruses carry DNA, and those are not awful because DNA is stable. It has lots of mechanisms to keep it reproducing or copying itself without mistakes or mutations. Because of that, it's easy to develop, or relatively easy, to develop vaccines for DNA viruses. So most of the viruses that have vaccines available, they're DNA based. However, RNA viruses have many replication errors because copying RNA is less precise than copying DNA. Oftentimes you have reverse transcriptase to copy that back into DNA, which increases the number of errors produced. So there are always new forms of it developing. So by the time you get one vaccine, there's a new variant, a new mix of DNA that vaccine won't work against. So things that are RNA based, like the flu and like HIV, you either have to keep kind of remixing or kind of changing your vaccine, which is why you need the flu vaccine every year, because the CDC adds the new strains or the new combinations that they're aware of to that flu vaccine. And things like HIV, where it's really hard or if not impossible to develop a vaccine because the virus mutates so quickly, any vaccines would only be functional for a limited time. Again, viruses infect plants, animals, bacteria. Pretty much everything is infected by viruses. Some viruses can only infect one type of host or one single type of cell. Others can affect a wide range. So rabies, it's a virus that can affect anything that's a mammal. Bats, dogs, humans, anything that's a mammal can be infected by rabies. What it's able to infect is determined by glycoproteins on the surface of the virus. So this image shows a virus and these kind of things sticking off the outside of the virus. Those are glycoproteins. If these can recognize the host or the host cell, then they can bind to the host and cause infection. Kind of like a key fitting into a lock. They can unlock the cell and get into it. New viruses can come from birds, so birds naturally carry a wide range of different viruses, and birds and humans are very different in their immune systems and everything else, so it is difficult to transfer bird to human unless you're in direct contact with a very sick bird. So direct contact allows it to pass. However, to get it into humans in a form or version that's more adapted to the human body, viruses will often go through a closer related species like pigs. So if pigs and birds are in close contact, or if the pigs are exposed to the bird droppings with the virus, things like that in there, then it has time to work out and establish itself in an immune system more closely related to humans. Then if humans are in direct contact with the pig, they can pass it to us. Notice this line goes both ways. So birds can get infected by pig strains, and pigs can get infected by human strains also, so that allows them to mix in the pig And eventually, if a virus is allowed to continue to mutate and adapt once it's going from pigs to humans, if it learns to jump human to human, now it can spread quickly through caretakers. If it becomes airborne, it can spread through the environment to anybody nearby. So that's kind of the cycle for new or developing emerging viruses. Goes from birds to pigs to humans, and then learns to jump human to human. HIV, and I again won't talk about this too much in the lecture because we will cover HIV thoroughly in lab. It's a retrovirus, which means it uses RNA and it changes that back into DNA using the enzyme reverse transcriptase. And reverse transcriptase makes lots and lots of mistakes and errors, which is why HIV mutates so fast we can't develop a vaccine for it. Treatments, they're aiming at preventing the HIV from attacking cells of your immune system. So HIV first infects something called a macrophage cell, and then it switches and attacks T cells. And those T cells are what controls your immune system. It's the base of your immune system. So treatment is focused a lot on preventing that shift and also just keeping the viral counts low so that it doesn't get triggered to make that shift. So just killing off the virus that are floating in your circulatory systems.